just want you to know Dr. Gerard Wagemaker is the last line on stem cell research. With setting up and doing laboratories all over the world, this is the key person. And all of you biotech, pharmacy people, this is the key person. Thank you.
And Commander.
It's not working. It's working.
It's working. It's working. Well, ladies and gentlemen, may I have my, here's my first slide. I cannot do it myself, I have to move it on, okay. Here you see the title of my talk. And here you see the spelling of my name, and the correct pronunciation is Gerard Wagemaker, but that's very difficult to pronounce for you, so you can call me Jerry. I have been working at Erasmus University in Rotterdam for 42 consecutive years, but I'm retired now, which in the Kingdom of the Netherlands means that you're kicked out. So I am working now at Hematopoietic University in Ankara, where I have a research lab associated with the Children's Hospital, which is the national referral hospital for rare diseases. And I'm also working in the Raisa Gorbachev Memorial Institute for Children Hematology in St. Petersburg in Russia, where I even have an office. And in addition, I'm affiliated with the Tulane National Primate Research Center in Covington, LA. Well, let me first say that I feel very privileged and honored to be invited to the ISGCT.India. And there are a few other things that you have to know about me. Firstly, I'm a totally humorless person, so I never make jokes. So if I make you laugh, it is by accident. Secondly, I am not on social media at all, so you cannot find me there at all, which probably means that I do not fulfill the criteria for being a social person, as has been explained to you in a few talks ago. Let's go to the business. Next slide, please. Great. It works. Fantastic. That was by accident. Well, ladies and gentlemen, in humans, we have identified so far more than 7,500 inherited disorders. And the genetic defect has been elucidated at the gene level. I have to make sure that I don't... Yeah, but okay. Has been identified in 40% of the cases, and that is rapidly increasing because the genome analysis is making progress very rapidly. Also, individually, those diseases are extremely rare. In total, an estimated 24 million people in the European Union, which is 6% of the population, are affected by an inherited rare disease based on a genetic defect. And unfortunately, for most of those, a curative treatment is not available, treatment being symptomatic, with adaptations in the living and work environments, and eventually, in many cases, intensive nursing and care. The healthcare costs are excessive. In my country, it is estimated that rare diseases, although nobody knows it exactly, consumes about 20% of all healthcare costs. In contrast to cancer, which is very well known and is very heavily subsidized by research, which is only 10%. Well, what can we do about it? If we know an inherited disorder occurs in a family, we can do preconceptional, prenatal or postnatal diagnosis, we can do genetic counseling, or whole genome analysis. we can give symptoms, so then we can try to prevent the birth of new brothers and sisters that have the same genetic defect. Or you can do it, or we can give, if they are born, symptomatic therapy. For some, and those are the diseases in which a certain enzyme is missing, and I come to that in a few slides, then you can try to replace it by the recombinant enzyme, so an artificial protein. But of course, the most logical approach would be to correct the genetic defect, and that is what we call gene therapy. I have been involved in the last approximately 30 years in the development of hematopoietic stem cell gene therapy, which is based on hematopoietic stem cell transplantation or bone marrow transplantation. And we have applied that to the inherited immune deficiencies. Perhaps most of you have heard from the so-called bubble boy, which was an American boy living in San Antonio, in the heart of Texas, so to speak, where the NASA developed a cover because he did not have any immune functions. And he survived that for approximately 12 years, but then of course he became an adolescent, broke out, needed a bone marrow transplantation, that failed. And I will also discuss particularly the lysosomal storage disorders, which are enzyme deficiencies that have very serious health effects. Of course, the most common ones are sickle cell anemia and thalassemia, which occurs very frequently in the Middle East countries, or occurred there very frequently. And we have approached that as well, but I won't discuss that. And another rather frequent rare disease is hemophilia, and we have approached that as well, and I believe we can now cure hemophilia as well. What am I going to do? I give you a short historic background, so you are correctly informed about the stem cell term and the development of stem cell transplantation. I go briefly to the primary immune deficiencies, and then give the example of a lysosomal storage disorder, which is a Pompe disease. I have skipped technical details about the gene transfer. I have also skipped ex vivo hematopoietic stem cell amplification, because you have MANA now. And I will finish with another type of a disease that we have started to approach as well. Some history. The bubble boy disease, severe combined immune deficiency, was first transplanted in Minnesota and in Leiden in 1968 and 1969. And that was done in the institute that I had the privilege to work in from 1974 till 1992, the Radiobiological Institute, TNO, under this guy, Van Beckham. And they published with their Leiden colleagues, first in 1968 and second in 1969, the first transplantation of bone marrow stem cells and fetal thymus in an infant with lymphopenic immunological deficiency. And it was the first time that such a bubble boy disease, severe combined immune deficiency, no T cells, no B cells, no immunity whatsoever, was successfully cured. And my laboratory was subsequently responsible for preparing those transplants. And in 1986, so that was some 15 years later, we published the first 162 patients transplanted in The Lancet. We did that together with 4 institutes, Leiden, Ulm, London, and Paris. Nowadays, we continued with developing bone marrow transplantation or stem cell transplantation for leukemia. And in 2014, we reached a total worldwide of 1 million patients treated, largely on the concepts that we developed in the 80s. Now, it would not be a surprise that when we had finalized that in 1986, we also started immediately to think about gene therapy, correcting the gene in the stem cells of the cell of the patients themselves. And that was pursued by two institutes, of those for London and Paris. What is the principle of gene therapy for such a disease? Well, you have a baby, the baby doesn't have any immune system, and you take some bone marrow, to be exact, roughly 1% of the bone marrow of that baby. Then you isolate the stem cells by a technology that is now commercially available, and then you have to get the right gene inserted in those stem cells. And we do that by what the Germans call the taxi, which is a viral vector, which is a vector, which is a retroviral vector that is capable, that is an RNA vector, that's an RNA virus that is capable of integrating into the DNA of the host. And originally they started with mouse leukemia viruses, or we started with mouse leukemia viruses. But in the meantime, we have changed it and we have perfected the technology. And now in an overnight procedure, so we close the lab. We alter the virus to the cells at 5 o'clock in the afternoon, go home, have a nice dinner with some wine, and then the next morning at around 10 o'clock, we look at the cells, and then 60 to 90% of the cells have the gene. Now, and then that is re-infused into the baby. And one of the interesting things of stem cells is that some people believe 100%, but I always say 60 to 90% of the stem cells find the way to the bone marrow themselves. So a bone marrow transplantation is not a complicated surgical procedure. You simply inject them intravenously, and 60 to 90% of the stem cells find their way to the bone marrow within 2 days. Of course, you have to make space in the bone marrow, and we do that by low-dose conditioning, but that's not necessary here. And then the cells that have the new gene start to differentiate and provide the right gene, and therefore the right protein and the right receptor to the T cells, the NK cells, the B cells, the monocytes, etc., etc., etc. So that's the procedure, and that's the procedure that is currently also used in the clinic. And we have really perfected it to such an extent that we can do that without loss of stem cells within 24 hours. We have developed this in European consortium that also has the original laboratories, the original institutes that did the original bone marrow transplantations for the severe combined immunodeficiencies. And we have, in the last 10 years, we have added Israel, because Israel has excellent science and also a lot of inherited disorders, and Turkey, because Turkey has the highest incidence of inherited disorders in Europe, which is due to the fact that they always have lived in very small communities, and therefore consanguinity has reached there a very high level. To give you an example, the children's hospital that I am connected with in Ankara is a hospital of 12 stories, 12 floors, 2,000 square meters each floor, and it is packed from the basement till the attic with rare diseases. So if you're interested in research on rare diseases, it is a paradise there. Right. Unfortunately, of course, it immediately went wrong in France, as usual. So also the research of the gene therapy were superior to the allogenic stem cell transplantation, that's the donor stem cell transplantation. both in efficacy as well as in overall survival, in 5 patients, nowadays in 6, the complication was the development of leukemia. And we rapidly elucidated the pathogenesis. The retroviral vectors that were used as taxi for the new gene had a propensity to land near proto-oncogenes, which resulted in aberrant expression of those genes and therefore in a pre-leukemic state. We have removed those vectors, except for one disease. We have removed those mouse leukemia viruses and have replaced them by the AIDS virus, of which, of course, all the pathogenic genes were removed, and used that also, that's a lentivirus, used that as the new taxi. And those vectors do not have the propensity to land near proto-oncogenes. And developed from 2002 to 2010, they are currently in clinical trial for Wiskott-Eldridge disease, which is an immune deficiency, for X-linked SCID, which is also an immune deficiency, the original one, for ADA-SCID, which is another, I come to that in a minute, and for two of the lysosomal enzyme deficiencies. Yes, I just told you that. So the AIDS virus-derived vectors are able to integrate into quiescent cells, which is another example, which is another advantage, which means that we can use a very short incubation time. We don't have to trigger the stem cells into cycle because the majority of the stem cells are non-cycling. We have deleted some of the regions that might influence neighboring genes, and they don't land preferentially near proto-oncogenes. Now the problem, of course, is that one of the problems is that your genome, to tell you the truth, is a fantastic chaos. It is an evolutionary chaos. It has 10% of retroviral vectors, retroviral viruses integrated from the past. And the consequence is that if you remove a gene from its, let's say, evolutionary place in the genome and place it somewhere else, which is what we do, it does not necessarily function very well. So we call that codon optimization. So we change the codons without changing the protein resulting from those codons to make it more easily transcribed when you place it in another setting. And the other thing is that the natural promoter, for the very same reason that drives the gene, that drives the expression of the gene, is not always optimal. So we have two things to do. We have, in most cases, to codon optimize the therapeutic gene, and we have to select the right promoter from quite a few promoters that we have available. And I will very briefly enter the development of lentiviral stem cell gene therapy for primary immune deficiencies now. This is due to this is due to the fact that I processed this an hour ago, this talk, on a Windows machine while it was produced on a Macintosh. Perhaps I can move on. Yeah, not bad. At the moment, let's say when we started, we only could distinguish in the 60s and 70s, we could only distinguish primary immune deficiencies with B cells or without B cells or double, but that was about it. Nowadays, we know approximately 134 different genetic mutations that result in a primary immune deficiency. 4 of those are the most important ones. One is the ADA-SCID, which is adenosine deaminase deficiency, which is essentially an enzyme deficiency which is systemic, but particularly affects the lymphatic system. The other one is gamma C deficiency. That is the receptor for one of the growth factors for T cells, the most important one actually, and that is also called X-linked SCID because it only occurs in boys because it is linked to the X chromosome. And the Y chromosome, of course, as you know from the behavior of many males, contains mostly nonsense. So that does not compensate for it. And then there are the recombinase enzyme deficiencies, the RECs, in the bottom of the slide. And those deficiencies are due to the fact that one of the two genes that you need to create the diversity of your immune response, your inborn diversity, does not function. I will very briefly go to one of those enzyme deficiencies to illustrate the principles. We have codon optimized it, and then we started to look at the promoter, and there we selected the spleen focus-forming promoter, which is a mouse leukemia, a virus promoter, which is the strongest promoter that we have. The own promoter, the REC2 promoter, the X-linked, the gamma C promoter, and a universal promoter, the UCO promoter, and that is about it, and compare that with the wild type. And then you can see that the UCO promoter, the green one, performed much better than the own promoter of the REC2 gene. And that determined that we are moving the gene therapy with the UCO promoter and not with the own promoter of the gene. To make a long story short, both for X-linked SCID and REC2 deficient mice, because I work in the preclinical evaluation, had a complete phenotype correction. Both disease entities are now ready for clinical implementation, and in London, the X-linked SCID lentiviral is now being in clinical trial. If you're interested in the details, the publications here stand below. Now I move briefly to the development of lentiviral stem cell gene therapy for lysosomal enzyme deficiencies. We have approximately more than 40 lysosomal enzyme deficiencies. A well-known one is the disease of Gaucher, which is a Jewish disease, but is also in other populations frequent, and Krabbe disease, etc. And we focused on Pompe disease for a reason that it was that we had in Rotterdam the Pompe Center. What is Pompe disease? Pompe disease is a deficiency of the enzyme acid alpha-glucosidase, which means that glycogen cannot be metabolized to glucose, because that's the idea about glycogen. Glycogen is the storage for glucose. And it's also called glycogen storage disorder, or glycogenosis type 2, or acid maltase deficiency, which is another name for the enzyme. And it is called by Pompe, by a Dutch doctor that described that some 100 years ago for the first time. So Pompe disease. Well, what happens when you cannot metabolize glucose? Then the glycogen accumulates in your body, especially in the muscles, which you see in the lower part of the picture. And the result of that is that the muscle fibers are being replaced by glycogen. And glycogen, as you know, does not contract, which means that babies that are born with this disease, within a few months, do not have any capacity to use a muscle because the muscles are simply not there anymore. We know several forms of Pompe disease, and that depends on the severity of the immune deficiency and the place of the mutation in the genome. We have the early onset, or the classic infantile, and those patients have developed rapidly with an enlarged heart. They get glycogen in the heart muscle, and they cannot function, they cannot breathe properly because their respiratory functions are impaired. And the most severe form, babies that are born with the disease, die within the first year. Here you see such a baby, which is already on respiratory support, and this is classic. If you pull arms, he or she cannot lift her head at all. There are, as I told you, many variants depending on the mutation and the degree of deficiency. The classic infantile to the late onset, and some people get it only diagnosed when they are around 40, which is quite extreme. And then that expresses as that they cannot move the muscles of their face anymore, which you see in the lower right panel. Okay. What is the current therapy? That was also developed at my university, at Erasmus University, in the 80s and 90s. Enzyme replacement therapy, which means that you give the recombinant enzyme at least every 2 weeks. And it is marketed under the name of Myozyme by the firm Genzyme. The problem is that it is a foreign protein, so certainly in the patients that do not make it themselves in any way, you get rapid immune responses, which means that you need more and more and more, and eventually the patients become refractory to the treatment. It requires lifelong administration, and the costs are extremely high. To give you an example of the cost, in the Rotterdam Pompen Center, the patients that are treated on average cost 424,000 euros per year. For the slightly more than 100 patients that are being treated there, the current costs are now 60 million euros. Which also means that to keep a Pompe patient alive for 20 years would require some 10 million euros. And there is a big discussion in my country and also in other countries whether that high price, in view of the value of lives, is worth it. Certainly because it's not fully effective. So there is an unmet medical need to develop a therapy, and that is why we developed, or we used the Pompe mouse model, which is here indicated as the GAA, that stands for alpha-glucosidase mouse model. And we do essentially the same thing as we do for, as I showed you in the human scheme. We take bone marrow cells from male Pompe mouse, from male Pompe mouse mice. We isolate the hematopoietic stem cells. We add the viral vector with the GAA, with the glucosidase gene, to the stem cells. We harvest them the next morning, as I told you, and then infuse them in a female mouse. And why do we use male mice versus female mice? Because then we have another criterion to measure whether how many donor type cells, how many repaired cells, how many gene repaired cells are being present in the recipient. And then in the same way, as I showed you for the severe combined immune deficiencies, the enzyme is being produced by the offspring of the stem cells. And as you're sitting here, you're making 1 million new blood cells per second. It is a tremendous factory of, when I teach to my students, I always say, well then, you do anyway something useful while you're listening to me. But that is a tremendous factory. And that factory is permanently renewed from the bone marrow stem cells by the mere fact that all the blood cells live very short. The neutrophils, most of the neutrophils live only for 12 days, for 12 hours. The thrombocytes for 8 days, the red cells for 3 months. And the replacement of those cells is the reason that you make 1 million new cells per second. So we have a tremendous endogenous factory here for the enzyme. Does that also work? Well, the muscles have the mannose 6-phosphate receptor, and by that receptor they can take up the produced enzyme by the blood cell system. What we have published some 7 years ago is that if you have 20 to 30% of the blood cells that express the acid alpha-glucosidase, it results in a high level production. It results in restoration of acid glucosidase activity in the tissues, and as a consequence, a reduction of the glycogen storage. We had a full correction in liver and spleen. We had a full correction in the heart, and we had an almost full correction, improved, but not fully normalized skeletal muscle function. But it was at least as effective as the enzyme replacement therapy. And the other thing we noted, and that is also characteristic, of course, for successful stem cell transplantation, is that we had a robust immune tolerance to the new protein. To move further, we optimized the protocol. We codon optimized the DNA sequence, the numbers of cells needed, the number of genes per cell, and we completed the safety evaluation. Now, I told you already that in many cases it is necessary to change the codons to make them more suitable to function. I will not enter the technical details. If you would like to know that, you can send me an email. And indeed, the codon optimized, that is the black bar, is approximately 20-fold more effective than the native sequence, which you can see here displayed. And then we looked, of course, whether that resulted in an improved efficacy, and that appeared to be the case. You only have to look at the black bars, which is with the codon optimization, the activity of the enzyme in the upper panel, and now you have to try to find the glycogen in the lower panel, which is the glycogen, and the glycogen there has entirely disappeared. Okay, so that was very successful, and then we looked at, we also looked at the function. We also looked at the function of the muscles, and indeed entirely normalized as well, but the slides are not in this series. And then also pompe does not result in mental retardation. There is substantial glycogen storage in the brain. And that glycogen storage, as we know now since a few years, is specific in the astrocytes. Because the astrocytes are the key cells that provide glucose and other things that are important in the energy metabolism of the brain. And the energy metabolism of the brain is one of the highest metabolisms that we have in the body, is exclusively done by the astrocytes. What you can see here is in the lower left panel that the enzyme level reached approximately half normal levels, but what you can see in the right panel is that it is almost completely normalized in glycogen. And that is what you also can see in the wild type, the knockout, and the enzyme and the gene therapy treated ones. And you see some residual glycogen. This was done at 3 months, and there's not completely steady state, so there is some residual glycogen. And for the rest, glycogen is always present in astrocytes, because it is the source for the glucose of the metabolism of the brain. And here you see that in the astrocytes, GFAP is specific for astrocytes. Green is the staining for the enzyme, for the, if you look at merged, then you see that the astrocytes, and that applied to approximately 100% of the astrocytes, it is loaded by the enzyme. How does it come there? We have published in 1988 that the monocytes, in case of brain damage or brain inflammation, are moving through the blood-brain barrier, and there cause an immune response. And in our case, we have demonstrated that indeed the monocytes, descendants from the gene-repaired stem cells, gene-modified stem cells, move through the blood-brain barrier and release the enzyme to the astrocytes, which indeed have the mannose 6-phosphate receptor, which is needed to take the enzyme up. So we had totally normalized skeletal muscle function. We had a reduction of glycogen in the brain, called quantitatively normal, astrocytes involved, likely mediated by the microglia, the descendants of the transduced stem cells. And this was reached with a very high viral copy number, and we are in the process of reducing that. A high viral copy number might have some safety risk because you don't know exactly where it is in the genome. But so far we haven't seen any adverse effects of the treatment in these mice. I'm skipping this one. Well, let's not skip it. The mannose 6-phosphate receptor is a quite weak receptor for the enzyme. And if we combined it in the vector with the IGF-2 receptor, which is in combination with the mannose 6-phosphate receptor much more effective in taking up of the enzyme, then indeed that is given in this scheme, then indeed that improved the situation considerably. And here you see a functional test, which is the so-called rotor rod. We place the mice on accelerating rods, and if their function is normal, they stay on the rods for approximately 2 minutes. But if their muscles do not function, then they fall off very rapidly. So the knockout mice indeed fall off very rapidly, but the codon optimized stayed on almost as long as the wild type mice. And that has improved further in the meantime. So we had a normalized skeletal muscle function, and we had a reduction in the brain, and with this trick of the IGF-2 receptor, an average viral copy number of only 1 per cell seems to be feasible. In the European context, we have developed that now for quite a few diseases. For Hurler disease, which is also a very specific lysosomal enzyme deficiency, for Pompe disease, for Fabry disease, which is also very costly. And 2 of them are in trial, and unfortunately I don't have the time to explain all those diseases to you, which is adrenoleukodystrophy, and a very successful trial is metachromatic leukodystrophy is now going on, which has been published in Science and a review recently in another journal listed below. I will close with another disease, stem cell gene therapy for mitochondrial disorder that we call MNGI. What is MNGI? MNGI is very complex. It has a very complex name. I can never remember it, but it's called mitochondrial neurogastrointestinal encephalomyopathy, MNGI in short. What is it? It is caused by thymidine phosphorylase deficiency. It is autosomal recessive. It starts between the 1st and 5th decades, and it results in progressive eye disease, loss of weight, peripheral neurological problems, diffuse leukoencephalopathy, so brain problem, severe dysmotility of the gastrointestinal tract, by which those patients cannot digest food anymore, and it's based on mitochondrial dysfunction because of accumulation of uridine, of thymidine and desoxyuridine. They go up. And here you see this patient. She was treated with a bone marrow transplantation, but her condition was that poor that she died from the bone marrow transplantation. Not by us, by the way, otherwise she would have died. To give you an example, we have 3 patients in Ankara with this particular disease, and 3 males, and they are around 18 years of age, and their average weight now is 25 kilograms. So that's an extremely disabilitating disorder. It's also a very rare disorder. There are only 70 patients described in the whole literature, and as a consequence, only very few are still alive right now. As I told you, it is due to thymidine phosphorylase deficiency, which results in accumulation of thymidine and deoxyuridine in the mitochondria and an imbalance of the pool. There have been therapeutic approaches that did not work, and of course we applied the same thing to gene therapy to the mouse model. And here you see the experimental setup. We used the knockout mice. They were sublethally irradiated. We did a biochemical, molecular, and phenotype analysis, and then we transplanted them for the second time. And what you can see here is that indeed, with one of the promoters that we tested, there was a very high expression in the stem cells. This resulted in a complete reduction of the a very high enzyme activity in the blood, which resulted in a complete reduction of nucleoside levels in plasma and urine. And the levels of donor chimerism were quite high in this particular case, and the average copy number per cell was only 2. And here you see that also in the brain, the thymidine phosphorylase activity was normal. And that was shown also in normalization of the brain. I'm not going into the details, but they have quite a few additional fluids in the brain, which were completely released by the stem cell, the gene therapy, which is also shown here in the myelin protein normalization in the right upper panel and the right lower panel compared to normal. Also in the tissues it was normal, and the summary and conclusions is that in principle we are ready for clinical trial here, except that the patients are that rare that it is very difficult to find money to treat them. So to conclude, I already discussed the diseases that are in clinical trial and discussed the ones that are in preparation for clinical trial. We have added now the mitochondrial disorder, and with this I conclude that clinical implementation of lentiviral stem cell gene therapy is well on track for 6 primary immune deficiencies, 5 lysosomal enzyme deficiencies, and 1 mitochondrial disorder. Long-term follow-up revealed a high level of efficacy without any severe adverse effects, both in the clinical trials and in the preclinical evaluation. Other developments are there, of course, also there. That is gene editing for some diseases. And efficacy and safety needs to be evaluated further. And so far, the current state is here summarized, and I've discussed most of it already. But so far, this approach is the only way to receive both a robust immune tolerance to the new protein and effectively bypasses the blood-brain barrier. This was done in collaboration with many institutes within Erasmus University and outside, and the MinG was done with Columbia University in New York and with the Universitat Autonoma de Barcelona, who was already autonomous before they declared independence. Thank you very much for your attention.
Thank you very, very much, Dr. Gerard. Jerry, I just want to sum up a little bit on what he was trying to say. See, there are some people with congenital defects and genetic defects. When there is a genetic defect, his research is paramount and has taken that genetic defect out, replaced it with another arena. This arena is going onto a viral vector. My question, Jerry, to you, do you understand? So he's taken out some of the cells, attached a viral vector to them, pushed them back into the diseased body, and in about 60% of the cells home into the target zones. He's making an arena place for those cells to go back as well. My question to you is, how do you make sure that that 60% cells are going back to the bone marrow?
Because we measure the chimerism, and we know exactly how many
Yeah.
because in all cases we measure the chimerism,
Right.
which we would like to keep around 30% max,
Right.
for the very simple reason that if in case something goes wrong, we always have 70% normal cells. Yeah, and in Canada they integrate something that they can kill the cells with the modified gene.
Okay.
But we measure the chimerism. We know exactly how many cells are left after the conditioning that we give. So we know that...
You're measuring the chimeral through what device?
The chimerism is measured by the FACS, the fluorescence activated cell sorter, which is done because we transplant male mice into female recipients.
Okay, so it's the Y chromosome with which you're distinguishing.
Yeah.
So you understood, he's taking it from the male, he's putting it into the female, and distinguishing which are the ones which has come in there, and so he's able to judge that 30, 60, 40% have come into the bone marrow. Clear? Understood? Now one more question. How did you change the virus vector? How did you put the good gene on it?
Oh, well, that's molecular biology. I didn't enter into that detail. I have a molecular biology staff that's doing the work.
Okay, okay. So
And-
is there an
and,
equipment or an enzyme or a culture plate, or what is it that actually does that work?
It is a standard. There's even a kit available commercially now. It is a standard 4-gene procedure, which is being done by specific cells that are capable of producing that virus. Yeah, because the retrovirus, of course, integrate into the genome to make new copies of themselves. So they automatically have the equipment to make
more
new virus.
Another question, and sorry, I'm interrupting on another important session here. One more question, which I wanted the audience to know, otherwise I can take you outside and ask you the same questions. But I want the audience to know as well. What is the reason that you chose the retrovirus for doing this work?
Because the retrovirus is an RNA virus that integrates into the DNA of the host cell to make new viruses. So
Perfect.
that is automatically equipped. And also the lentivirus, the HIF virus, is a retrovirus in that respect. Yeah. So you have different types of retroviruses. Many of them are mouse leukemia viruses. I worked with them already 40 years ago. And the HIV virus is an ideal virus if you remove the pathogenic genes, because it has all the capacities of a retrovirus in what we need. And in addition, it does not have the propensity to cause leukemia. Yeah, it is doing something else as it's in its own job. It does not cause leukemia. So that was an ideal step, and that was actually done in California by a guy there, and it was moved to Milan. And in the European context, we developed it further.
Thank you very much, Dr. Gerard Wagemaker, and I bow my head in appreciation and honor to the work that you have done all your life. You have taught us so much on gene therapy, on transfer of genes, chimeral work. It's amazing the body of work that you have done all along. Thank you very, very much.